Midterm exams can be so tough. I've been there before myself. There are so many topics that have been covered and these questions sound so weird. So in this video, we're going to go through some difficult essay-based questions your professors could ask, and we're also going to break down some multiple choice questions that could be about the same topics to prepare you for your midterm exam. I myself have given some of these questions before, so you're going to be very well prepared. Hey everybody, Organized Biology here, where we make difficult biology concepts simple. And today we're going to start with cells and tissues. So there's a question, explain the process of diffusion and how it contributes to gas exchange in the body, and include how concentration gradients are involved. Great. Okay, so first there was that term called diffusion. You definitely need to know this term. Diffusion, okay, is simply shown by two symbols, high to low. We're moving things from high to low. So for example, say we have some sort of a membrane here, okay, like a cell membrane, we'll talk about it in a second, and we have high amounts of, say, sodium on one side of the membrane, and then we have relatively low amounts of sodium on the other side of the membrane, okay? Well, in this case, we've got what's called a concentration gradient, where we have a relatively high amount of something on one side and a low amount of something on the other. Now, for most cases, sodium will only worry about sodium. So even if, for example, there was like high amounts of calcium over here, just as an example, we're really not going to worry about the amount of calcium when we're talking about sodium diffusion. So again, diffusion is things moving high to low, and I'm going to clarify down their own concentration gradient. That basically means they're only going to worry about themselves. Now, there are exceptions to that rule, but for your teachers, they're likely going to teach it the way I am showing it right now. Now, that being said, I want to give a quick question for you. Can sodium pass directly through the cell membrane? Can sodium, or calcium for that example, can it pass through the cell membrane? Well, the answer is actually no, because the cell membrane is made of a non-polar molecule, primarily lipids, okay? And we know that anything that is charged, like calcium, like sodium, even like water, it's a polar molecule. All of these things, again, are something called polar molecules. That means within themselves, there's a separation of charge. Like in water, you don't really need to know this from my class, but there's a separation of charge where the oxygen is slightly negative, it's a little electronegative, hydrogen is slightly positive. So therefore, separation of charge within the molecule can't pass through nonpolar things. Same thing with calcium and sodium. They're ionized, they are charged, and that just basically means they're dissolved in water, and they also cannot pass through the nonpolar membrane. 99% of the time, they will need something called a protein in the membrane that will basically allow that sodium to diffuse in. If we diffuse these things through a protein, we call that facilitated diffusion, because the protein is facilitating the diffusion of sodium across the membrane. Now, in the other case, where would calcium want to go? Let's say there's a calcium protein, a protein that's going to allow calcium to move high to low. Where would it want to go? Well, in this case, calcium wants to go that way, okay? So that's the role of concentration gradients in diffusion, when we have a high concentration, low concentration on two sides of a membrane, and we flow high to low. But the question wasn't about these guys. It was about gas exchange, right? How gases are exchanged in the body. Now, we know we breathe in some gases in our lungs. So I'm going to draw the lungs here, a little cardiac notch there. So here's your lungs. Okay, and the lungs, if you were to zoom in on the base unit, at the very base, and then throughout the lungs, what we would see is something called an alveoli. So we'll see these alveoli, these are little gas sacs that are lined with a specific type of tissue. So one of my questions could be on a test, what type of tissue is lining the alveolar membrane? So again, this is the alveoli. Okay, what type of tissue is lining the alveoli? We see flat, thin cells. These are called simple squamous cells, okay? Simple meaning one layer, squamous meaning basically squashed and flat. Therefore, in the alveoli, if, say, we have a lot of oxygen in here, which we would in the alveoli, okay? If we have a lot of oxygen in here, and there was another tissue type butted up right against it, the bloodstream, okay? So here's the bloodstream. Now, fun fact, the bloodstream is also lined with, in the capillaries specifically, very thin, tiny little blood vessels. They're also lined with simple squamous. So why are we doing that? Why are we putting simple squamous cells at the base of the alveoli and in the capillaries, the small blood vessels lining them? Well, we know these guys are very, very important for what? Diffusion of things. Think about it. If we have a very thin layer of something, it's very easy to get moving things in and out, right? So as an example, oxygen's really high in your lungs, and at this point, you don't need to learn about the cardiovascular system yet, but we know that in the blood, oxygen will be very low because we have just delivered it to all the tissues. So if there's high oxygen on one side of a membrane, low oxygen on the other side of these membranes, where will it flow? Obviously, high to low into the bloodstream. That is how we get oxygen into our blood to go to all the cells of our body, by using simple squamous cells in diffusion. Now at the same time, we know in the bloodstream, we will have a high amount of another molecule, another gas, called carbon dioxide. Now, carbon dioxide will get here and it will say, hey, what's the gradient? Well, in the air outside of us, there's very, very little CO2. So I'm just going to draw like one. Okay, there's one molecule of CO2 over here. So you guys know if CO2 is high on this side, low on another side, what does diffusion say? We're moving high to low. CO2 leaves, okay? And then CO2 is actually forced out when you breathe out, you exhale, and CO2 leaves the body, which is amazing, okay? So that's how diffusion happens at the lungs. Now that being said, in your class, we know that we're talking primarily about the cells. Now the cells of the body, you have 30 trillion of them. They're very, very needy. They need to get a blood supply all the time. So once we have packed full the blood with oxygen, that oxygen will travel down the bloodstream, go all tissues of your body, and we will just show one of those cells of your body right here. Let's just say it's like a fibroblast in your skin or something like that. Well, we know these cells are constantly doing a specific process called cell respiration, where they're taking a molecule of glucose, they're combining it with a gas of oxygen, specifically 6 of them, and they will produce our ATP molecule. That is the key. We've seen that earlier, right? ATP needs to be used for muscle contraction as an example, right? So we use those two things. We get oxygen from our air, glucose from our diet. We throw them in our bloodstream and we feed our cells. So how does that happen? Well, oxygen will be in the bloodstream, right? High amounts. And oxygen will actually pass high to low into the cell. Now you may say, well, Mr. J, you told me that things can't really pass through the membrane very easily. But we know that oxygen is actually nonpolar as well. So oxygen can just diffuse right across the membrane of the, again, simple squamous cells of the capillaries, as well as the membrane of your cells, okay? Glucose is a little more complicated. Glucose is a polar molecule, so if I were to draw glucose here, it's a pretty big polar molecule. There will be proteins in the membrane that will facilitate, again, the movement of glucose from high to low into the cell as well. So that's where we get glucose in. But this is about gas exchange, right? That's what the question asked. So we've got the oxygen going into the cells, but in the same process, we know in cell respiration, we also produce 6 CO2 molecules, if I can draw a C, as well as 6 water. The 6 water will just kind of hang around wherever it needs to go, but the CO2, we know, leaves. CO2 will build up, build up, build up in the cells, and it will go from high to low into the bloodstream, okay? So all of a sudden, we're packing our bloodstream full of CO2. We're basically feeding all the oxygen to your cells, and now we're high CO2 here, low oxygen in the blood. Where does that go? Back to the lungs, where we now get the CO2 out, get the oxygen back in, and the process continues on and on and on for the rest of your life. So that's how diffusion of gases occurs, both at the lungs, oxygen flowing in, CO2 out, and then at the tissues of the body, where oxygen's going into the cells, CO2 is going back into the blood, and then we do it over and over again. Very well done. So a couple questions we could ask here is, hey, what is the relative concentration inside your cells compared to the bloodstream for CO2? What's the relative concentration of CO2 in the cells in the blood? Well, we know there will be a relatively high amount of CO2 in the blood, in the cell, if I can speak right. High amount of CO2 in the cell, low amount in the blood, so it'll flow high to low out of the cell into the blood. Now, another question could be, why can CO2 and oxygen diffuse across membranes easily? Well, because I mentioned these are non-polar. Any gas typically is going to be non-polar. So I'm going to write gases here as well. So gases can just pass through those membranes very easily and get into and out of the cells. Awesome. So those are a couple questions you may see on your test. The muscular system. So outline the sliding filament theory of muscle contraction, including the roles of actin, myosin, troponin, tropomyosin, calcium, and ATP. Now this is a very multi-dimensional question, y'all. So we are in the muscular system, and we're talking specifically about the sliding filament theory. Now what you need to know is the sliding filament theory is going to be only in skeletal and cardiac muscle. The reason for that is these two muscle types look virtually the same. They're these long, thin fibers, these muscle cells, and they have what we call striations. So these striations, in order to contract, are going to actually come closer together. So when the muscle cell contracts, obviously all those lines will come closer together, and that is how we actually contract the muscle. It's actually very similar to an accordion. If you've ever seen an accordion played, you can stretch them out and then come closer together with those lines. So the question is, what's happening here, and what are all those proteins doing? Well, if we were to zoom in on the muscle tissue, it's really easy to draw actually. You will see a thick filament that is called myosin, and you will also see a thin filament called actin. So I'm going to draw actin as this blue. So here is actin, here is myosin. Okay, now first off, anytime you see IN in an A&P class, it means it's a protein, and it was produced by the cells during protein synthesis. We'll get to that later on. Now actin is actually attached to something that is a little zigzagged, and that is called our Z line. So we're going to have this Z line here, okay? It's some tough connective tissue, so it's bound to that actin. So as you can see, the myosin is going to act on the actin. That's how I remember it. Myosin literally means muscle protein, and it is going to grab that actin and pull it towards the middle. This middle is called the M line. It doesn't actually exist, but it is just an imaginary line right down the middle of that main myosin head. Okay, now that being said, myosin is also on the other side. So there'll be myosin heads over here. And if we obviously grab the actin on both sides, pull it towards the middle, we're going to pull these Z lines together in a very similar way. We're going to pull all of those striations of the muscle fiber together. Now that being said, that's only actin and myosin. Myosin is going to grab actin. But we need to be able to control when we do that, when we actually contract those muscles. So we're going to zoom in here, and we're going to see a few different components that we did not see on this image above. First off, we're going to see actin, and it's actually going to have some individual circles. These are actually the individual actin proteins. They look like little circles. They're also actually in your cytoskeleton, fun fact. And again, they are attached to that Z line. We can draw that again. Okay, but on the actin, we're going to have a new protein called tropomyosin. This is a very long named protein. I'm going to point to him. Tropomyosin. Okay, so I remember tropomyosin, long name. It's a long protein that's actually covering the actin. Okay, now bound to tropomyosin, we will have another protein that will be above it in a way. We're going to draw it kind of like a little receptor. Okay, so this is called troponin. I want you to think of troponin as the lock, okay? We're gonna have a key that's gonna go into the lock at some point, okay? So we got troponin bound to the tropomyosin, covering the actin, and then obviously we're going to have those myosin heads that are going to be coming up as well. So here are the myosin heads again attached to that big myosin protein. Okay, so what's going to happen here? Well, we know myosin's going to grab the actin, try to pull it inward. But the problem is tropomyosin is actually covering, right? This is covering the binding sites on the actin. They're called the myosin binding sites. Binding. I cannot speak and write. If you've watched any of my other videos, you know that to be true. Covering myosin binding sites on the actin, okay? So therefore, if it's covering the actin, the myosin basically cannot grab the actin. So this is at a point where the muscle is not contracted. So here's a multiple choice question that a teacher could actually ask you on a midterm final. Hey, when tropomyosin is covering the myosin binding sites, what is happening? Well, you could say there's no muscle contraction occurring, because if we're covering it, we cannot bind myosin to actin, therefore we cannot contract the muscle, okay? So that's one question you could be asked. You're welcome. Now that being said, when we want to contract the muscle, we need to have some sort of key that goes into the lock that will actually pull this chain off. Think of it as a bicycle, interestingly. So say you chained your bicycle around a pole and you locked it closed. Well, how do you get on the bicycle to ride it? Well, you have to insert the key into your lock, you pull the chain off, and now your strong myosin legs can bind to the pedals of actin. It's a great analogy to think of this, like a bike ride, okay? So we need a key for the lock. What is our key? The key for contraction is calcium. I always remember that. Calcium is the key to contraction. So there will be an ionized form of calcium come in, and that's going to bind to that troponin. When we do that, the tropomyosin is actually going to get pulled off of the myosin binding sites of actin, and it will actually pull about right here. So I'm going to kind of draw it moving around over here. Okay, so we're pulling that chain off. Notice in A&P, anytime something binds, here's another good question. When something binds in A&P, what happens? Well, usually something moves or changes its shape, therefore it changes its function. So here we have calcium binding troponin, pulling chain off, something's gonna happen. What's gonna happen? Well, now we have a mosquito about to bite me. I got it, I'm safe. It's about to take my blood. So myosin can now bind with actin. So I'm going to draw it kind of coming up. Okay, here's myosin binding to actin now. And that myosin, once it binds to actin, is going to execute what we call a power stroke, where it's going to pull the actin to the left. So all of this would shift over. So basically think of myosin head grabbing it and then pulling it like that. Myosin is that muscular head moving the actin this way, and that is what executes a muscle contraction. And again, that is called a power stroke. So one question could be, hey, when myosin pulls actin, what is that process called? It's called a power stroke. Awesome. Now, that being said, you notice that the myosin moved, right? Myosin moved. This is a protein that is literally moving. Well, how did that happen? Well, enter in our friend ATP, okay? So in order for this myosin head to work, we need ATP. But what's kind of confusing about this is more like a catapult than anything. Have you ever used a catapult before, where you basically load something up in the catapult, you push it back, and then all of a sudden, boom, it fires off, right? Well, we noticed that the myosin here was already kind of cocked back, and then it can pull the actin. Well, in order to get from this relaxed state into that energized state where we're pulling that myosin head back like the catapult, we have to use our friendly molecule ATP. So when ATP is cleaved, it goes to an ADP and a phosphate group. That ADP and a phosphate group actually, interestingly, bind to that myosin head. Now, whenever you see this in anatomy and physiology, it just means we're inserting some sort of currency of energy. When ATP is used, something cool happens. Well, what is that? Well, we're taking a relaxed myosin head and now we're putting it into an energized head so that, boom, we're energized and we're prepared to grab actin and pull it whenever calcium binds to it. Okay, so that's the role of ATP, to re-energize the myosin head. It's a good answer to a question. So that when calcium binds, tropomyosin pulls off, we can contract that muscle. Now, that being said, there is a step before this. I said the calcium just appears, right? When did calcium appear? And that could be a question they ask you on this test. When does calcium appear in the sliding filament theory? Well, it appears in response, I'm going to say in response to basically a signal from a somatic neuron. Signal from a somatic neuron. The somatic neurons are these motor neurons that can communicate directly to your muscle tissue and basically excite them to contract. What would that look like? Well, there's a long process called the neuromuscular junction. I'm going to simplify it. The neuron comes in here and it's going to communicate with that muscle fiber, right? That muscle cell. So therefore, we have something called a neurotransmitter. I'm going to draw it as like a triangle, okay? That neurotransmitter is released, binds to a receptor on the muscle cell. It's actually called a ligand-gated sodium channel. You don't need to know that. And eventually, that creates some sort of intracellular processing that will increase the amount of calcium in your muscle cells. Therefore, calcium binds and all of this happens, your muscles contract. So the key here is the neuron triggered the muscle contraction, triggered the calcium being present, and it happens when you want to contract your muscle. You can actually control these neurons like I'm doing right now. I'm controlling my neurons, telling these biceps to contract. Absolutely fascinating. But again, the ion important for this is calcium. So if they were to ask, hey, what's the direct ion that helps induce muscle contraction? It could be, is it sodium, potassium, calcium, or chlorine? Well, you would say calcium. It's the key to contracting. By binding to troponin, pulls the tropomyosin off, and then myosin binds to actin, pulls it, and then we re-energize the myosin head with ATP, and we can keep contracting and contracting and contracting, making those power strokes. Awesome. So a lot of questions that could go into that, but all through that long essay-based question about all of the roles of the proteins and ATP and calcium. All right, so now we're into the skin. So I apologize for getting under your skin in this one. Pun intended. Anyway, a patient presents with a third-degree burn on their forearm. Explain what layers of the skin and underlying tissues are damaged and discuss two major clinical concerns for this patient. Okay, this is a great question. I can definitely see you getting this one on your test. So we know we're talking about the skin, okay? And the skin is the body's biggest organ, interestingly enough. So we've got, there's another mosquito in here. I gotta go kill it. Give me a second. Saved. Okay, so we got 3 layers to the skin. On the top part of the skin, I did not plan that. On the top part of the skin, we have something called the epidermis, okay? That stands for upon the dermis, which is the main part of the skin down here. Okay, so this is the dermis, derm meaning skin. Now, a couple things with the skin. Epidermis is going to be stratified squamous epithelial tissue. So you've got a lot of flat cells lining the body. Okay, and then you're going to have these basal cells on the bottom that are continuously regenerating. They're stem cells. They're going to be pushing these cells upward to eventually their death. Okay, now the reason you have this stratified squamous tissue is to protect the underlying tissues of your body. It makes sense, right? Now the dermis is going to be actually dense, irregular connective tissue. And that basically just means, hey, there's a lot of collagen fibers throughout the dermis, okay? Very strong, tough, kind of stretchy a little bit. And there's going to be some cells, okay? Cells, gels, and fibers consist of connective tissue. These are fibroblast cells. They're going to be putting down the collagen, basically keeping that skin tough and connected. Now, underneath it, we'll also have a layer of what we call the subcutaneous layer or hypodermis. You may hear either one depending on what your instructor says. So I'll write them both. So it could either be called your subcutaneous layer, or it could also be your hypodermis. All right. And as I drew this is primarily made of just adipose tissue. It's a type of connective tissue consisting of fat cells. These are basically just a bunch of fat cells. They're very big and quite literally fat, and they're made of fat. So why do we have skin? Well, it's for protection primarily in the epidermis, okay? It's going to also have like hair follicles in here, sweat glands in here so we can thermoregulate. We have the sensory neurons that'll be wrapped around hair follicles to detect things. So there's a lot of functions of the skin. But in the problem, we're talking about a third-degree burn. Well, we know with burns are classified based on depth. So a first-degree burn would be, hey, we burned down through the epidermis, but we haven't gotten into the dermal layer. So again, this is a first-degree burn. Then you can also have a second-degree burn where you're into the dermis, and that is going to be a problem. Why is that? Well, we know in the dermal layer, we have a blood supply. In the epidermal layer, there really wasn't a blood supply, so now we've exposed the blood to the outside environment. Not good. And further, in our question, we have a third-degree burn. So we have burned into the subcutaneous layer, again, third-degree burn. And so now we've got the fat tissue basically compromised, and everything below the skin, y'all, this is what becomes a problem. There are muscles underneath here. There are bones underneath here, other connective tissue, a whole lot of very vascular, high blood supply tissues. So, what have I mentioned twice? Blood supply. If we have a burn down even second all the way to third-degree burn, we are now exposing the blood to the outside environment, exposing blood. And if you know about blood, it is mostly made of water. Okay? Your body's made of about 60% water. Now you are basically exposing that water to the outside environment. What's the worry here? Well, if you have a glass, I don't have a glass of water up here, but if you have a glass of water and you pour it onto a table, what happens after like an hour? All of that water will have evaporated, right? So if we expose the blood to the outside environment, we will lose a lot of water. So the number one clinical connection and clinical concern will be dehydration, which makes you think, hey, what's a function of the skin? Well, it's to insulate the body, keep the fluid inside of the body. So you can think, hey, what's the function of the skin? Insulation. You're trying to prevent water loss, okay? But then a second thing is we know the blood basically goes to every tissue of the body. And what really likes the blood supply is bacteria. So if we've exposed the blood to the outside environment, we can also have a great risk for infection. These are the two biggest concerns anytime a person gets a second or third degree burn. And so therefore, infection, particularly with bacteria, will be treated with, as you can probably guess, antibiotics. Okay? So two biggest things when you're treating a burn patient. This could be a question on a test. What would you do to a burn patient with a second or third degree burn? Number one, you'd likely give them an IV. Number four, basically rehydrating them. So you're going to be giving them some fluids through an IV. And then secondly, you're going to be giving them some antibiotics, very wide spectrum, wide ranging antibiotics to try and prevent any bacterial infection that could occur when you expose those tissues to the outside world. Great question, and a lot of questions that we get asked about it, right? All right, before our next one, if you've been enjoying this video so far, consider liking the video, subscribing to the channel. You can also check out the shop. We've got some cool shirts, some coffee mugs that have been ordered, so enjoy, use it at your leisure. So the next up is about the nervous system. So explain how neurons detect stimuli, send action potentials, and include the roles of sodium, potassium, as well as neurotransmitters. Okay, this one's evil. If your professor does this one, this is like a 2-hour lecture that they would give you. So we're going to summarize it here in about 10 minutes. So first off, we're talking about neurons, okay? So neurons are going to be the functional cell of the nervous system. So if we look at any one neuron, typically it'll have three main parts. We will have these little detectors, I like to call them. These are going to be called dendrites, okay? So I always remember that dendrites detect. Dendrites detect. So they will have, amazingly, these little receptors that can detect a variety of stimuli. We'll talk about them here in a second. But then it'll be connected to the main cell body. This main cell body is going to be called the soma. Okay, this is a typical, like anytime you see a cell, you see the nucleus in the center and the cell membrane on the outside. The soma is just basically the main cell body of the neuron. But then the most important part of the neuron, I would say, will be this long end called the axon, okay? The axon is this big long end, right? It can be very short. It can be like microns long, like millionths of a meter, or it can be actually up to about 3 feet long in some areas of your body. And this is going to be that signaling branch. Okay, so we're going to be sending a signal down the axon, but we remember axons will send action potentials. Action potential is a fancy word for basically how the neuron sends its signal. So there's a lot to break down here. But again, we need to know first how the neuron detects different stimuli. So if I were to zoom in on any one of these dendrites, usually what we will see is the following. So I'm going to draw kind of like a membrane of sorts on the dendrite. Okay, so imagine this is inside the cell, okay? This is outside the cell. This is also outside the cell. And this is going to be that membrane. Now, obviously, I've made some breaks in here. These breaks are going to show different protein types. So we're going to have one protein be this guy right here. We'll make it a little channel, and he'll have some receptors. We're also going to have this guy. Okay, it's also going to be a gated channel, and he's going to have kind of these little levers. So I'm going to have this. Okay, so what is happening here? Well, again, the dendrites is going to detect a stimuli. Okay, so what types of stimuli could we, quote unquote, detect? Well, if you take your finger and maybe poke at some of your arm hairs or maybe poke at your skin, you feel that, right? You're detecting that stimuli. What's the stimuli? It's just mechanical force, right? So we need a way in this dendrite to kind of detect or have a receptor for some mechanical force. So this guy right here, this specific type of protein in some sensory neurons are called mechanically gated channels. Mechanically gated channels. That's a fancy word for saying, hey, if there's some sort of mechanical force on the channel, it will open up. So imagine we put some force on here. We put some force on these little levers of this protein. Again, it's a membrane protein. The cell produced it through protein synthesis, put it in the membrane. We put a force on those little levies, and what would occur is that protein would amazingly open up. Okay, it would open up. And that's going to do some cool things here in a second we'll talk about. So just know, force opens up this protein. The second one is going to be something called a ligand-gated channel. Ligand-gated channel. Now that's a fancy word for basically saying it's a lock and key mechanism. Lock and key mechanism. What do I mean by that? Well, there is a lock right here, a little receptor. I'm just going to say lock. And we are going to have some sort of key come in and bind to that lock. Typically, these little keys are just chemicals. Okay? A lot of the times, fun fact, they're neurotransmitters. We'll see that here in a second. But those chemicals, again, can bind to the lock. Okay? And then we can open up that gate. We'll draw this gate again. All right, we're opening up the gate when that key is in the lock, and therefore we can allow some things in. All righty, so those are just two different types of how we can detect a certain stimuli. One can be force, one could be a chemical like a neurotransmitter or even a hormone. Okay, so what's happening here? We're opening these gates, but what's going to occur? Well, we need a little help with our understanding of action potential. So first, let's come back to the neuron. One thing I want you to know about any cell of your body, including your neurons, is they have a relatively negative charge inside of them. Okay, why is that? Well, inside of your neurons, you have all these cool looking proteins inside of them, as well as all of your cells. And these proteins, okay, that your cells are producing constantly, are always going to have this strong negative charge. Okay, so that's what accounts for the negativity of your cells. So really, we're all just very pessimistic because our cells are very negative. Terrible joke. Okay, but moving on. So we're negatively charged. If we were to literally like take a voltmeter and measure the charge inside of this neuron, it would be about negative 70 millivolts. That's kind of cool. That's kind of cool. Now, different cells will have different negative charges to them, maybe like negative 90, negative 50, but we're just going to say negative 70 as an average. Okay. Why do you care about that? If the cell is negative, I want you to know it is doing nothing. If the cell is negative, it is doing nothing. Now, that is an overstatement. It's not doing nothing. It's like making proteins, making energy, all this stuff. But in terms of sending signals, getting action potentials, it's doing nothing. Okay? So, that being said, if we have a negative cell and we want to send a signal, like if this neuron needs to communicate with somebody, well, we need to go from negative to positive. So I want you to write somewhere that if we get positive, if positive, we will send action potential. If positive, send action potential. In fact, an action potential by definition is a flow of positive charges. So that could be a question you have on the test. What is an action potential? When a cell goes from negative to positive, and those positives are sent down the axon of a neuron. So it would look kind of like this. Positive charges getting over here. Things flow from high to low. Those positive charges would flow down all the way to the end of the neuron. In response to that positive charge, just to remember this before we get into it, when that positive goes to the end of the neuron, this is called the terminal end of the axon or the synaptic terminal, whatever your professor uses, there will be chemicals, interestingly enough, inside of little vesicles, little capsules inside of the cell of the neuron. And those vesicles will spew out that chemical. And that chemical, fun fact, is called a neurotransmitter. So I'm going to write that, neurotransmitter. Now, those neurotransmitters are, again, chemicals that are released by neurons that will communicate to some sort of target cell. So we're going to write here target cell. And that target cell could be a variety of things. It could be, say, a muscle. We've talked about that earlier, where the neuron talks to a muscle cell to tell it to contract. It could be a gland, okay, like your salivary glands. Maybe you start getting hungry, you send a signal telling yourself that you need to start salivating. And you could also be another neuron, okay? So neurons can communicate with neurons. Like in your brain right now, neurons are like going crazy, kind of trying to understand this information, right? So those are just target cells that can receive the information from the neurotransmitter. But how does that work? Well, the target cell has to have receptors for it. So, target cell must have receptors for neurotransmitter in order to respond to it. Another thing with neurotransmitters, they can either stimulate the target cell, okay? So they can stimulate the target cell or they can actually inhibit the target cell. We'll see that in another question when we're talking about parasympathetic, sympathetic. So that's just an overview of a neuron. Once again, it's negative. If it gets positive, we send a signal, release a neurotransmitter to a target cell. We communicate with somebody and tell it to do something, either stimulate it or inhibit it. But the question was, at the very beginning, what are the roles of these guys? How do they come into play with getting from negative to positive? So that's where we need to draw something new, okay? So we're gonna draw something new here, where we know, here's a neuron. I'm gonna draw a neuron, okay? And it's just gonna have this big box to it. And that neuron is going to have, again, its axon, okay? So let's say this is a sensory neuron and it has that dendrite, okay? And that dendrite is going to detect that mechanical force, right? So we open up that mechanically gated protein channel because there's some sort of force on the sensory neuron. Okay, so there's a force there, and we open these guys up. Well, fun fact, these mechanically gated channels are called mechanically gated sodium channels. Okay, mechanically gated sodium channels. So they're going to allow sodium to now flow in or out of the neuron depending on the gradient. You remember talking about the gradients earlier. But the thing is we know that in neurons there is this amazingly important protein inside of the neurons called the sodium-potassium ATPase pump. Sodium-potassium ATPase pump. If you were to get a question on a test about the sodium-potassium ATPase pump, you will say it's the most important protein in the membranes of all cells. That is true because, interestingly, we know it runs on ATP energy. So whenever we make a molecule of ATP, we cleave it up and we make ADP and a phosphate. Okay, so we're using energy in the process of using this pump. Fun fact, about 50% of your calories is estimated to go towards firing this one pump in all of the cells of your body. Pretty crazy. So a lot of the food you eat goes to firing that by making that ATP. Now, what does it do? What does that pump do? Well, it's going to pump, throw things low to high, sodium out of the cell. So sodium will be constantly pumped out of the cell. Whereas potassium, we'll draw in, we'll just do it in orange. Potassium is going to get pumped into the cell. So if we do that all the time, what we're making here is what we call a gradient, where now we have a high amount of sodium outside of the cell and a high amount of potassium inside of the cell. That is true. Now, why does that help? What's the point of that? I never understood this when I was in your shoes as a student. Well, we know the cell is negatively charged regardless, right? We're doing nothing. But what if we were to open sodium channels? We know sodium's high outside the cell. Where will sodium want to go? Well, sodium will rapidly flow in, yes? So now we're gaining sodium into the neuron. Now I'm going to race a little bit above here. If sodium, in response to that stimulus again, were to go into the cell, what charge will the neuron slightly become? Well, it'll get a little more positive. So let me kind of draw this as a step-by-step process for you. We've got a force acting on this sensory neuron. Sodium begins flowing in. Well, what was the charge of the neuron at the beginning? -70. So here's the graph of the voltage. Okay, so here's the voltage of the neuron over time. We were at -70, were we not? Okay, so we're at -70, but then one happens. A force opens up some sodium channels. Sodium flows in. What happens? Well, we're going to go a little bit higher, are we not? Because sodium is flowing into the cell. Positive charge is flowing in, making the cell slightly positive. Well, at a certain point, specifically -55 millivolts, -55, we know this is called the threshold potential. You may have heard of this before. The threshold potential is basically when we open up a boatload of new protein channels. I'm going to draw them up here. This guy is going to be called a voltage-gated sodium channel. I kind of draw them like Harry Potter foreheads, right? These are voltage-gated sodium channels. Well, think about it. Voltage-gated. They will open and close at different voltages. What are they going to allow in? Sodium. So at this point, -55, these suckers, which you have a ton of them, will fly open. So now, if we open up a bunch of these at -55, sodium is rapidly going to flow in. I'm going to draw a big arrow flying the sodium in. what will happen to the neuron's charge? It will get positive like we wanted it to, right? So we can communicate with somebody. So sodium flies in, cell gets really, really positive, and what will the graph look like? Boom, action potential. Goes up to about positive 10 millivolts, which is crazy, okay? So we got this cell, got really positive. Well, let's think about it. When we get really positive, all of those positive charges that were coming in here because of the sodium will flow from high to what? High to low, which we saw earlier, right? It got positive here, sent it down the axon. So all those positive charges are going to flow down what we call the axon hillock here, all the way down to the end of the neuron. When we get here with those positive charges, we know what was at the very end. These little vesicles, right? These vesicles contain neurotransmitter. The neurotransmitter is going to be inside the vesicles. Now, at this point, very key point here, I'm going to add a new ion here. If the question on a test were, what does calcium do for a neuron? Here's where it comes in. We're going to have voltage-gated calcium channels at the very end of these neurons. Voltage-gated calcium channels. Again, Harry Potter, voltage-gated calcium channels. One thing you need to remember is that calcium is always high outside of the cell as well, very similarly to sodium. So if calcium channels open up, so we'll just draw them opening, calcium will now flow into the neuron. Calcium, we said earlier, is the key to contraction, but I want you to remember even further that calcium likes to move things. Remember, calcium likes to move, bind it to your opponent and pull tropomyosin off. In this case, calcium loves to push things. Like Patrick with Bikini Bottom, pushing it somewhere else. We're pushing the vesicles out, allowing that neurotransmitter to be released and communicate to whatever target cell we're talking about. So how did that happen once again? We were negative. We opened up the channel, right? Sodium flows in. At a certain point, threshold potential, negative 55, we open up voltage-gated sodium channels, flows in, gets super positive, right? That super positive sends it down the axon, opens up other voltage-gated channels, pushes pulls calcium in high to low, vesicles with neurotransmitter pushed out because of that calcium. Amazing. That is the goal of inaction potential. But here's the thing. I haven't mentioned, really, potassium. What's the role of potassium here? That's the last part of this question. Well, we know, fun fact, if cells were to stay positive, so let me just draw this, you don't have to. If the cell were to stay positive for a long period of time, the cell actually induces apoptosis, into basically programmed cell death. We don't want your neurons to die every time they send one signal. So what we need to do immediately after getting positive is actually get back negative to protect the cell. So what happens here? Well, I want you to know that at this point, at positive 10 millivolts, we're going to, oh, by the way, I can kind of draw number 2 here, was this whole crazy thing, right? All these things happen at the same time at threshold. At the top part though, at 3, I'm going to give you a list of things that happen, okay? Number 1, your sodium and calcium channels close. Okay, sodium and calcium channels close. That is very important. So all of these guys, voltage-gated calcium, voltage-gated sodium, will close back up. So I can kind of write number 3 here. We're closing at that action potential. We're closing these suckers back up, okay? If we do that, we no longer have calcium, sodium flowing in. But at this point as well, we will have voltage-gated potassium channels opening. Voltage-gated potassium channels open. So at this point, let's draw it over here. Here's a voltage-gated potassium channel opening at 3. Okay, so we're going to draw 3 here. Therefore, potassium is high inside of the cell. Where will it flow? Out rapidly. We have a lot of these voltage-gated potassium channels. So therefore, if we are no longer gaining sodium and calcium, but we're losing a positive, what will happen to the graph? It will go down very, very quickly due to voltage-gated potassium channels opening. So, couple questions that we could have here. What's the role of potassium in action potential? Well, the role is for potassium to flow out after the cell gets positive to get the cell back negative again. What was the role of sodium? Well, it was to enter into the neuron, thus getting it super positive to send that action potential and release the neurotransmitter with the help of calcium, right? But then what's depolarization, repolarization? You'll probably get these questions. Well, the Depolarization process is when we're getting positive. So I'm going to write on the left side of the graph, depolarization. This is when we're going from negative to positive. But then we repolarization, due to potassium. So you can remember that. Repolarization is going from positive to negative again, or just further negative, okay? So that's the repolarization process. Now, after all this crazy stuff happens, what do we need to do? We've just lost a lot of potassium. We've just gained a lot of sodium. We need to fire that pump again, do we not? Right? Think about it. We've just gained a lot of sodium, lost a lot of potassium, but we want potassium high inside. We want sodium high outside. How do we do that? We use the pump again. Isn't that amazing? So once again, we bring sodium in, send the action potential, release neurotransmitter. Then we let potassium out, and then we have to reorient it with the sodium-potassium pump. So the question could be, hey, after an action potential, what reorients the concentration gradients? We will say the sodium-potassium ATPase pump, okay? So many things go into this one question, but I really hope that you've seen several different parts of it. So let me ask you a couple other questions. What is the role of, say, calcium at the end of a neuron? Well, it's to push vesicles of neurotransmitter out of the cell. When does the sodium-potassium pump come into play? After the action potential is sent. What's the role of negative 55 millivolts? We call that, again, the threshold potential. And what if the question was, what happens at the threshold potential? Well, we opened up voltage-gated sodium channels. So there's a lot of questions that could go into just this one concept of action potentials and ions. All right, we're going to stay in the nervous system for a second, then we're going to hop back to cells, tissues, and chemistry. So the next one is explain why damage to the myelin sheath in diseases like multiple sclerosis affects nervous system function, and then give the roles of the sympathetic and parasympathetic divisions when regulating heart rate. Okay, so kind of two different questions here about the nervous system. So let's start first with the myelin sheath. We actually didn't mention that just before, but we have our neurons, right? With that soma up here, okay, here's the soma, and then we had the dendrites that we talked about a little bit, and then we've got the axon. Well, the axon I mentioned could be very, very long. So in very long axons, fun fact, we will have these sheaths covering the axon, and these are called myelin sheaths. So I'm going to label these as myelin sheaths. Now, myelin sheaths, I want you to think of as are like insulation, okay? Because think of neurons as like power lines, right? Or even like cables of different things. So if you have like a long cable, do you know that like when you look at a cable, let me just hold one up for you real quick. Okay, if you see this cable here, do you see the wire in the cable? Well, actually not. You don't see the wire, you see the insulation. Because the insulation helps that signal travel faster than if it was just a bare wire. So same thing with the neurons. We're sending these very fast action potentials, negative to positive charges. They fly so fast, we're going to use these myelin sheaths, their insulation, to speed up the action potential. Okay, now one question you may get on a test is, hey, what produces the myelin sheath? Well, it's kind of a trick question because it depends on where it's at. If we are in the central nervous system, this is basically the brain and the spinal cord, you will have cells called oligodendrocytes. Oligodendrocytes. Okay? Oligodendrocytes will be basically these glial cells, and the glial cells are literally called the glue cells, and they're going to actually form the myelin sheath in the central nervous system. Again, the oligodendrocytes do that. But then we also have in the peripheral nervous system, that's basically all of your sensory and motor divisions. Okay, so whether you're sending the sensory information back to the brain or spinal cord or sending information out from them, this is going to be done by something called Schwann cells. Okay, so the Schwann cells are basically the same exact thing, but for the peripheral nervous system. They look a little different as well, but they will basically form that myelin sheath. And then you could also ask the question, hey, what is the myelin sheath made of? Well, if it's a cell wrapping itself around, it's actually basically the membrane of the oligodendrocyte cells or the Schwann cells. Okay? So basically, think of this as an extension of the cell membrane wrapping around the axon, insulating it with that layer of cell membrane, fat essentially. And there's going to be some other proteins in it as well. So again, what's the role of the myelin sheath? Speeds up the action potentials. But in the question, multiple sclerosis is basically an autoimmune disease where you've got your lymphocytes coming in here, these little white blood cells, and they're going to begin attacking that myelin sheath for some reason or another. If we were to damage that, it's almost as if you are fraying the insulation on a wire. Well, in that case, there's a chance that if the insulation were damaged, well, now that action potential may not be sent as properly. Just like if you have some tearing of your insulation in a wire, the electrical charge, rather than flowing all the way down the wire, it might trip out and go somewhere else, which is not good. So you can expect some pain, some burning, some numbness, and then some motor function loss as well in multiple sclerosis. All right, moving on to the parasympathetic, sympathetic, and regulating heart rate. So let's start with the heart, okay? The heart has a very important tissue that you haven't learned about probably yet in your class, but it's called the SA node. So I'm going to point to this guy. This is the SA node. This is just basically going to be the pacemaker of your heart. So it's going to be basically sending signals constantly as you feel your heart beating throughout the heart, making the heart positively charged to contract every second or so. Now that being said, this is an automatic function. So that's one question you could get on this test is, hey, is cardiac muscle voluntary? Is it voluntary or is it involuntary? Well, cardiac muscle tissue is involuntary because we cannot consciously control the contractions. It happens automatically. Happens automatically, you say, Mr. J. Well, we know that's likely going to be basically conducted by something called the autonomic nervous system. Autonomic nervous system. I want you to know that this is a branch of the motor division where basically any motor neuron is going to be talking to a cell and telling it to do something cool. So like move a muscle, talk to a gland, stimulate the heart muscle, etc. So it's going to be acting on a cell, changing its function a bit. To contrast that, you also have the somatic motor system that controls your skeletal muscles. So the autonomic is going to be controlling primarily internal organs that you cannot control. Now, how does it do that? Well, it's got two main branches. One branch will be originating, I'll draw, here's your spinal cord in your brain. One will be originating in the thoracolumbar region, so T to L vertebrae, and it will be your sympathetic nervous system. Sympathetic nervous system will come in and talk to those internal organs, but at the same time, in your craniosacral region, so in your brain stem, as well as in your sacral vertebrae, okay, I should be saying sacral end of your spinal cord, we will have the parasympathetic nervous system, where we'll have parasympathetic neurons coming out and communicating to different internal organs. So that's one question you could get. Hey, where would a sympathetic neuron originate? Well, you would say in the thoracolumbar region of your spinal cord. Okay, now in reality, the sympathetic parasympathetic neuron system is actually a two-neuron system. So for example, it would actually go from here to another neuron. And then for the sympathetic neuron or nervous system, it would actually go from here to something called the sympathetic ganglion right about here, and then go to the tissue. So one question you could get is how many neurons are involved in any parasympathetic or sympathetic response? It's always going to be a two-neuron system, a pre-ganglionic, post-ganglionic neuron, meaning before a ganglia and then after a ganglia. Okay, a lot of questions they could ask here. I'm trying to prepare you for both. Now, what the main part of the question was, is how do they interact to affect heart rate? Well, we know if the parasympathetic division is activated, okay, I'm going to write parasympathetic. Parasympathetic is always going to prepare your body to rest and digest. So it's the basically calming section of your autonomic nervous system. So in this case, we're going to release from this neuron some inhibitory neurotransmitters that will bind to the SA node, and that will slow your heart rate. Okay, so anytime you're in a calm state, parasympathetic nervous system is basically acting more, and so therefore it will slow down your heart rate. Whereas vice versa, the sympathetic division down here is going to prepare your body. That was a weird M. Look at that M there. Sympathetic is going to prepare your body for fight or flight. So it's going to amp you up like during exercise, during a stressful scenario, and it will actually send stimulatory neurotransmitters. In that case, the SA node, the pacemaker cells, detect that stimuli, and it's actually going to speed up your heart rate. Speed up. So I like to think of the autonomic nervous system as basically like a little angel and devil on your shoulder. They're constantly talking to the same thing, but they're basically exerting opposite effects. The devil tells you to do one thing, the angel tells you to do the opposite, right? Same thing with these two divisions. They're talking to the same thing, but exerting opposite effects. Now, to clarify this, what if we were talking about, say, the intestines? So let's say we have our intestines down here. Alrighty. If we have our intestines, and we have, again, internal organs we're going to affect from these systems. What if the sympathetic system came in here? And what if the parasympathetic system came in here? What would either do to the intestines, you think? That would be a great question a teacher could ask you. Well, we know parasympathetic is rest and what? Digest. So interestingly, these are going to act as stimulatory neurotransmitters to the intestines. Whereas on the flip side, if you are in fight or flight, you don't give a crap about digesting right now. So you're actually going to have inhibitory neurotransmitters that will affect the intestines, slow down the movement of the intestines, whereas the parasympathetic will actually speed up the movement of the intestines. So it's not always one always stimulates. It could be different depending on which organ you are talking to. All right, next up is describe the roles of the membrane, nucleus, and mitochondria in maintaining homeostasis within a cell and use some specific examples. Oh, the amount of questions we could ask about this one. Okay, so with any cell, we are going to see some main components. Here's the outside of the cell. We know this is the cell membrane. On the inside of the cell, usually we have a nice sized nucleus here. So this is going to be that nucleus. Okay, this should be review. And then lastly, for a lot of the cells in your body, we'll also have some small thin organelles that look like this, and these are going to be your mitochondria. Alrighty, mitochondria. Now, the reason I actually drew them all in the same color is because each and every organelle, as well as the membrane itself, is going to have really a membrane around it. If you were to zoom into the cell membrane, you would see the following structure, which is really important in maintaining homeostasis. So we're going to zoom in here and look inside that membrane. We would see some little circles on the top and the bottom. And then we would see these long extensions on the base of each of them in two different layers. This is called the phospholipid bilayer, the phosphate group being the circles. I'm going to write P here, and then the lipid group here. Now, because the majority of the space of the membrane is going to be a lipid, we treat the membrane as a non-polar lipid. Now, that being said, you know that your cells are bathed in fluid. There's fluid all throughout your body. So therefore, there's likely going to be some water chilling outside of your cell as well as inside of your cell. So there's going to be water here. Well, therefore, how does the membrane keep water in one side and the other while still interacting with it? Well, we know that the phosphate groups here are going to be polar, just like water is also polar. So they can kind of interact with each other. But then we've got a big layer of fat in the middle to separate out the intracellular fluid from the extracellular or outside fluid. So that's one thing you could get on a question. How does the parts of the membrane help the cell contain two different areas, outside and inside the cell? Well, part of it's lipid to separate out the watery portions, and then the phosphate groups can interact with the water, keeping it on those separate sides. All right, cool. Now that being said, that's one aspect of the membrane, but we've seen, right, earlier, we could have some proteins embedded in the membrane, right? So we have these proteins that could potentially allow things in and out. So these are considered membrane proteins, alrighty? And as an example of a neuron, whenever we need to send an action potential, we can open them up and allow some things in and out. So a lot of the times, the membrane proteins can facilitate something called, I can't spell while I'm talking, facilitate cell transport really well. Okay, specifically for, as we've mentioned before, polar molecules like glucose, maybe some amino acids. So here, let's get an example of this real quick, because that's one of the questions. Let's say if the cell needed amino acids. Okay, so we have amino acids out here that we ate. We know amino acids are the base unit of proteins, okay, the macromolecule that we need to build our proteins. So here's some amino acids floating out in the bloodstream. I'll draw them as like triangles and squares. We know that there are 20 different amino acids. Think of them as little Lego blocks on how to build these proteins. And those amino acids will leak out of the bloodstream, okay, through these things called fenestra. You don't need to know that. And therefore, we need to get them inside the cell so we can build the proteins. How do we do that? Well, a lot of the times we will have these proteins that are called either uniport proteins. We'll say this is a uniport protein that will basically bind to a specific amino acid. So let's say this one comes in here, and it will transport it, in a one-way unidirection. So it'll basically pull that amino acid through so that we can bring it into the cell. So that is one way that we can facilitate homeostasis through these membrane proteins, bringing them in. And then again, we can build these together, put them together, chain them together through a process of protein synthesis, and we could form proteins like these guys. Very cool, okay? Now the question is, if we get those amino acids, how do we know how to put them together? Well, this is a long process. We won't go into the details of protein synthesis, where we have DNA inside of the nucleus. We will transcribe that DNA onto a molecule of what we call mRNA, messenger RNA. So the information stored on mRNA will go out to a structure called a ribosome. It's not actually this big, but ribosomes will read, we can remember reading RNA, okay? And it'll read that mRNA, and in the process of reading that mRNA, we will know, hey, let's grab that amino acid, and that amino acid, and that amino acid, putting it together, basically like a recipe. You have the recipe, you got the ingredients, and you put the ingredients together, okay, to form what we call a protein, okay? And again, how does that protein maintain homeostasis? It could be a membrane protein that will facilitate the movement of things into the cell. That's a great example. Another example was maybe we have actin proteins, okay, we talked about those earlier. They actually form something called the cytoskeleton. Okay, that as well as something called tubulin, another protein, and they hold the cell up, basically keeping the membrane the right shape so that it can keep the right function. So that again is how we can maintain homeostasis in certain cells as well. But moving on to the mitochondria, we know the mitochondria are mighty, right? Mighty mitochondria because they do something called aerobic respiration. It literally translates to with oxygen breathing. But whenever we're talking about cell respiration, it's talking about specifically ATP production. Okay, we know ATP is the functional unit of energy for the cells. I like to think of it as the currency of energy. So if we make this, we can do all the cool cell functions like making proteins and moving things to membranes and all sorts of stuff. So how do we do that? Well, we know we need glucose and oxygen. So we add glucose and oxygen, go through 3 steps, glycolysis, Krebs cycle, and then the electron transport chain in the mitochondria. You can learn about it more on my channel. And that mitochondria will spit out that ATP. It'll also spit out, as we mentioned earlier, some water. And then also in that process, we'll also make some CO2. We know that CO2 goes in the blood, oxygen comes back into the blood with glucose and all sorts of stuff when we keep making ATP. But again, the goal of mitochondria is to make massive amounts of ATP for all sorts of intracellular functions. So a question could be, in which process is glucose used for a cell to maintain homeostasis? Well, we'd say glucose is in the process of cellular respiration, cell respiration, and we know that that produces ATP, okay? What's a role of ATP in the cell? Well, we know the ATP can power the sodium-potassium pump, right? And we saw that earlier, so that a neuron can function well and all cells can function well. We know that ATP is also used whenever we're making proteins, adding proteins together. So we know if we need proteins for the cells, we need ATP to do that, okay? So that's a lot of things that we can do with it. What is the membrane, polar or nonpolar? Well, primarily nonpolar, but it has polar ends to interact with water to keep it intracellular and extracellular. All sorts of things, right? There's a lot of questions I could ask about it. Hopefully those were a few good ones that you may see on a test. All right, so here's a chemistry and cells question. Describe the structural organization of macromolecules and give examples of each of them in the human body. This is a great one. The one topic that I keep going over and over again in my classes is about macromolecules, so it's important that you know these. So we know the 4 macromolecules are going to be carbohydrates. So carbohydrates, they're going to contain carbon and hydrogen and oxygen, so that's why we get carbohydrate, interestingly enough, like carbon and then hydrate, water, H and O. Then we have lipids, and then we have our proteins, main one, and then lastly, nucleic acids. Now, for 3 of these, this guy, and this guy, we're going to organize them based on monomers and polymers. What do I mean by that? Well, monomers are going to be single units of these 3 things. So as an example, the monomer of carbohydrates will be monosaccharides. Monosaccharides, these are things like glucose and fructose, etc. Now, what that basically means is that for a carbohydrate, this would be a monomer of glucose. I'm just going to draw it as a hexagon, C6H12O6. So that could be a question on a test. Is this a monomer or a polymer? This is a monomer of a carbohydrate. But usually, most of the time, when carbohydrates are found in your body, they're not in that base form. They're actually in these chained forms. So sometimes we can have chains of glucose, bound usually by an oxygen, so it could look like this, okay? In this case, we have many units of that sugar. So we call this a polymer, poly meaning many units, right? So as an example of carbohydrates, this would be a polymer, and we know that a polymer in your body is something called glycogen. Fun fact, glyco always refers to carbohydrate, and then ogen usually means inactive or not used yet. So in your body, let's say we have cells, okay, and this is true. This is your liver cells, okay? And a lot of the times in your liver cells, we will have this glucose coming in frequently. I drew that one terribly. Look at that hexagon. I didn't pass geometry class, as you can tell. So if we're in a state where we have plenty of sugar, so for example, you eat a high carbohydrate meal, you jam-pack your blood full of glucose, well, a lot of the times we have too much glucose in the blood that we actually need. In that case, the glucose finds its way into the liver cells called hepatocytes, and they'll actually combine these suckers together and form our glycogen molecule, right? So we now have our glycogen molecule. This is basically stored carbohydrates for later. If we have no glucose or very low glucose in our bloodstream, we can actually break this down, release it into our blood, and maintain our blood sugar. So that's a great way we can kind of decide, hey, do we want things in a polymer? Do we want to store it? Or do we want to break it down and release it into our bloodstream? So that's one example of a carbohydrate monomer, base unit of energy, and then polymer, we have stored energy. So based on the different structures, we'll have different functions for them as always. That's anatomy and physiology at its finest. Now for lipids, we don't really have monomers, polymers for lipids. If your teacher were to ask, the base monomer would be something like a fatty acid or a triglyceride, but we're not going to touch them. Proteins, though, do have monomers, polymers. We've mentioned them before. We have amino acids as our monomer, amino acids. And you need to know that there are 20 different types in your body. So when we eat food, okay, that has protein in it, we break them down into each individual amino acid. And we can just draw amino acids as different shapes. Okay, so all these different amino acids are going to have obviously different shapes. And so if we chain them together, say we put all these guys together, we would form something called a polypeptide. Polypeptide means many amino acid units, and we call this polymer simply a protein. Okay, and we've seen proteins in this class, right? We've looked at membrane proteins, we've looked at cytoskeletal proteins, we've looked at actin, myosin, tropomyosin. All of these are proteins, and fun fact, they usually end in IN. Now with proteins, okay, they are the structure and function of all of your cells. So we've seen them before, but I want to add one more, and that is going to be a protein called an enzyme. Enzyme, I want you to think of it as either molecular scissors or staplers. These are molecules that can either break things apart, so basically take something and chop it up, or we can take an enzyme and we can staple things together, like we saw with glycogen, right? There was an enzyme that stapled these glucose molecules together to form glycogen. So enzymes are scissors or staplers, and you will usually hear on a test, how do enzymes work? How do enzymes work? Well, let me just give you an example real quick. If I had, say, this, let's see here, this long string here, okay? This is actually a cord that I have, okay? And if I wanted to, say, cut this cord in half, I could pull all day and work really hard to try to break this cord, but it's never gonna happen. However, if I take, say, this knife, this is actually a letter opener, and if I were to do this, I could just basically move it up and down and it would cut through that wire really, really quickly. Okay, so what did this knife, this enzyme, do for me? Well, it made this process of cutting the cord easier. The way we describe that is the enzymes lower activation energy. They lower activation energy. That is basically saying the energy needed for some chemical reaction to occur. The energy needed to tear that cord apart. That enzyme made it easier, lowered the amount of energy I needed to invest to break it apart. Okay, so that's how all enzymes work. But again, they can act as scissors or staplers. Now, in your tests, a lot of the times, a teacher will just say, hey, what does this, and this is a made up enzyme, there's a different name for it. What does this enzyme do? What does this glycogenase enzyme do? Well, you know, ACE always refers to an enzyme, okay? And if it's by itself, if that ACE is by itself, we are going to have an enzyme that cuts, okay, breaks things apart. But then it'll tell you what it's breaking apart. What are we breaking apart? Glycogen. So this would be the little scissor, the little protein enzyme that is going to be cutting apart the glycogen molecule. Now, what if instead we had an enzyme called this, glycogen synthase? Well, anytime you see synthase or the following two, polymerase or transferase, these are all going to be enzymes that are going to act as those staplers. They're going to put molecules together. So we could assume a glycogen synthase enzyme would be one that takes the monomers of glucose and chains them together to form our glycogen. Okay, so that's a helpful hint for suffixes and prefixes to know what the enzyme does. So that's a very common thing you'll be asked on a test. Now the last one, nucleic acids. Nucleic acids, the monomer of nucleic acids will be nucleotides. Nucleotides, if you need to know it, will always have a phosphate and a sugar as the backbone. Okay, so phosphate sugar backbone, it's actually a pentose sugar usually, so it's a pentagon. There we go. And then there will be a very important part of it called the nitrogen base. I'm just going to write N base. This nitrogen base is probably the one that you're going to be worried about because it has your A's, T's, G's, and C's, or U if it's RNA. Okay, so nucleotide is basically one of these letters, and most of the time in nucleic acids we have polymers. The polymers of nucleic acids will be our DNA or our RNA. Okay, you may hear of other ones as well, but they are basically when we have these long chains, so imagine the sugar phosphate backbone with a lot of letters. So whether it's A, G, T, C, etc. Okay? So again, we're chaining these guys together to form the polymer. And the functions, obviously, these are storing genetic information. So genetic info. And we know that the genetic info tells us how to code for our friendly macromolecule proteins, because it'll tell us essentially, hey, which amino acids do we put together in order to form that proper protein? So again, DNA, we transcribe it onto mRNA. We saw that a little earlier. That mRNA goes to the ribosome. We attach the right amino acids to build the polypeptide protein. Okay, so these are the main macromolecules that you see over and over again in your cells. So you need to understand them in order to understand how the whole body operates as a whole. So some simple questions you could be asked is, hey, what's a monomer of a nucleic acid? Well, you would know that that is a nucleotide, right? Well, what's involved in a nucleotide? Well, we know we have a carbohydrate, interestingly enough, a sugar, phosphate, and a nitrogen base. So you could say, hey, in a nucleotide, what atoms are represented? Well, we know that there's a phosphate group, so there's a P. We know in sugars we have C's, H's, and O's. And in the nitrogen base, we have nitrogen. So CH and P are all represented in the nucleotides, the nucleic acids, okay? Now with lipids and carbs, we know both of these will contain C, H, and O. Carbohydrates typically have more O's than the lipids, all right? Another question you could be asked is, hey, which has more energy, glycogen or glucose? Which has more energy, glycogen or glucose? Well, we know glycogen has a lot more atoms bound together. Glucose has very little, although it's used directly for energy. This one would have more stored energy because it has many, many more bonds, okay? What if you had a lipase? What's a lipase do? Well, lipase would be an enzyme, again, ACE by itself always means enzyme that cuts, okay, in this example. Lipase would tell you that it is a lipid cutting enzyme. So there's a lot of questions that could be asked about this. Best of luck. All right, this is a great one to finish on. It's an integration question that could, man, have a ton of little multiple choice involved in it. So you eat a high protein meal, trace the path of amino acids from digestion to absorption and how they could be used to build muscle tissue. Oh, I love it. This literally brings everything together. So you eat a high protein meal. So I'm just going to designate you eating it as this tube, okay? So here's your mouth going down to your stomach, all righty? And then we're going to have our intestines, okay? So here's going to be some of the intestines. Okay, so now we eat a high protein meal. So we're going to designate proteins just as this big square. So here's our proteins here. And that is a macromolecule, right? We get them from other animals and plant sources, and we throw them into the tube that is our digestive tract. Now the proteins, okay, right now are inside or outside of your body. I always confuse my students by saying this is actually outside of your body, because if they were to continue through the tube of your gut, eventually they would just end up as poop, and they are still outside of your body. So what's really weird is the inside lining of your digestive tract is technically outside of your body. But your goal is to get the protein, specifically the amino acids, to all parts of your cells, every single 30 trillion one of your cells. How do we do that? Well, we have to get it inside of our body. That is always and forever going to be your bloodstream. So I want you to remember, inside of your body is technically your bloodstream. Outside of your body is technically the inside lining of your digestive tract. So what do we do here? Well, we have proteins that are made of individual amino acids. So imagine all of your food are already in their protein form, their chains of amino acids put together. So what do we need to do with these? In order to absorb the amino acids by themselves, so they're small enough, we have to chop it up. We have to chop up all our food. That's called digestion. So in our stomach, in our intestines, we will have these scissor enzymes, okay, that are going to be called proteases. Proteases. These are things we'll hear about later on, like pepsin, like chymotrypsin. These are all enzymes that are going to chop apart these proteins. And hydrochloric acid is actually going to help out. We're not going to get into that. So in the process, we're going to chop up all of these proteins and turn them into individual amino acids. We'll just draw them as these shapes. From there, we will have a variety of different protein channels embedded in the intestinal cells. So imagine these cells lining the intestines. These cells, I'll zoom in on them. They are columnar cells, fun fact, and they will have a variety of different protein transporters that will allow those amino acids to pass through them directly. So they'll pass into the cell, and then the cell will actually pass them out through uniports into the bloodstream lying right underneath it. So we've got, again, proteins involved that these cells made to allow those proteins to be facilitated and move all the way through themselves into the bloodstream. Super cool. Okay, so now we have the amino acids in our bloodstream. Well, what did we need to feed? Our muscle tissue. So this blood is obviously going to go everywhere. We know the blood is a river of life, travels everywhere, and that includes your muscle cells. So imagine here your muscle cells, okay? And we know those muscle cells have those sarcomeres, right, with the myosin, the actin that we've talked about earlier. Okay, so these proteins are basically chains of amino acids put together in the form of myosin, actin, all that stuff. As the amino acids come through the bloodstream, again, the muscle cells will have to have transport proteins that will allow those amino acids into them. So they'll diffuse into the muscle cell. And so now the muscle cell has its base unit of proteins, amino acids. Well, you know that there's nuclei inside of the muscle cells, and that will be constantly throwing out some mRNA, okay, into the muscle cell. Let's say this mRNA codes for something like myosin, the protein myosin. Well, in that case, that mRNA would transport to the ribosome. It would be read by that ribosome, and the ribosome would put together these amino acids in such a way to form our myosin protein. And then, obviously, we'll read it for actin, and tropomyosin, and troponin, and we'll put all these proteins together to form our happy muscle unit. It's called a sarcomere. And now our muscle can be able to contract. So that is how we go from food absorb it into blood, travel everywhere, get to muscle cell, build those amino acids together into a polypeptide, something like myosin, and then we have our structure and function of our cells. Wonderful. All right, that's enough for today, but in order to prepare properly, I didn't mention chemistry as much, so I recommend going to my basic chemistry for A&P video, as well as my comprehensive cell biology video. But if you want to do other exam reviews, I've got Unit 1 and 2 on lock for you as well. Best of luck on your midterms, and go A&P.